Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Hi, everybody. This is Jason Key at SB Grid in Boston. Thank you for joining what is our final webinar of the webinar season. So this will be our last one before the summer. We're still setting up the lineup for the fall. So if you've got software you want to hear about, developers you'd like us to reach out to, people you'd like to recommend or put on the webinar list, or if you've got titles of your own software you're developing that you think people would like to hear about, reach out, let us know, and we can see if we can fit you into the schedule. We're putting that together. We'll be starting in September, and those run through the entire academic year through June. So we're trying to fill in at least one a month. We usually do at least one, but sometimes more. And let us know what you'd like to hear. With that, it is my pleasure to welcome Genki Terashi from Purdue, so my undergrad alma mater, and which would be his second webinar for software titles. He's coming to us from the Kihara Lab, and the Kihara Lab has developed a number of useful and interesting titles, almost all of which use deep learning or machine learning, AI functionality to address structural biology problems. And today's talk is no different. He'll be telling us about diffmodeler and cryo-read, macromolecular and nucleic acid structure modeling for cryo-EM maps using deep learning. So Genki, go right ahead.
Thank you for the nice introduction. So today I'm going to talk about the two tools, so diffmodeler and cryo-read. So diffmodeler is a macromolecular structure modeling tools. Then cryo-read is a nucleic acid, DNA, RNA structure modeling tools. So both tools are using the deep learning. So I want to talk about the diffmodelers, the overview of diffmodelers, the algorithm. Then also I show the diffmodeler tools on SB Grid, the command, how can we use the diffmodeler on SB Grid. Then what is the output files looks like. Then also how can we see the intermediate files. It's interesting files. A lot of interesting data is available. Then also I talk about the DNA, RNA structure modeling, so cryo-read, the overview of cryo-read, how the cryo-read program works. Then also I will introduce how to use our tool cryo-read on the SB Grid package. Then command lines and output files, then how we can interpret the result files. Okay, so before start the algorithm part, so I want to say that's the kind of situations, the background of this development. So as we know that the number of the data itself is rapidly growing. So for example, the cryo-EMs. So last years, they have a new 9,000 map data. In total, EMDB, the EM map data is over 45,000 entries. In the PDB, protein structure data bank, there are already 200,000 entries. Then every year, over 6,000 atomic structure models are deposited. Then recently, the DeepMind, Google released the AlphaFold protein structure database. So this database contains over 200 million entries. Then also, AlphaFold server is available. So everyone can easily generate the atomic model from the servers. And also it's very accurate. So in the AlphaFold server, every day, user can generate the 30 models. So that means that we have a lot of data, lot of cryo-EM maps, and a lot of the atomic model, experimental atomic models, and predicted atomic models. Then suppose we have this kind of the relatively intermediate low resolution map. Then to interpret this map data into the atomic models, maybe we can use the AlphaFold. First generate the individual chains or complex structure, then try to fit these atomic model into the density map. Then after fittings, we try to find the best possible combinations, then assemble the entire structures. So currently, this is a common way to build the atomic models from the intermediate or relatively low resolution maps. So to do that, to address this problem, so because the fitting and assemble the protein structure itself is not easy, so we developed the diffmodelers. So diffmodeler is using the conditional diffusion models. So by using the diffusion models, so backbone positions are enhanced from the input original density map. So this is for example, the input EM map, so relatively low resolutions. Then we train the network using the deposit PDB structure, ground truth PDB structure. So from the completely noisy data, the network learned how the backbone positions look like from the completely noisy data and using this EM map, original map as a conditions, then try to remove the noise iteratively. Then finally it gets the backbone position enhanced predicted density map. So these diffusion models are working very well, especially the relatively low resolutions. Even the helix structure, helix position looks like almost cylinder or tube structure, but sometimes the diffusion models can enhance the helix pitch, that we can clearly see that the helix backbone positions and also kinked regions. So this is very helpful to build the very big entire atomic models from the intermediate or low resolution EM. So this is the overview of the diffmodelers. So diffmodeler is a combination of the traced backbone positions by using the diffusion models and rigid body model fittings by Vesper. So Vesper is our rigid body fitting tools. So suppose we have a density map, also the sequence file. Then from the density map, so as we saw that we can trace the backbone positions using the diffusion models. Then from the sequence, we can use different protein structure modeling tools like AlphaFold or AlphaFold database or ESM-Fold, like so on. Then once we generate the structural pools, then diffmodeler automatically fit these individual chains into the enhanced backbone positions. Then we have a lot of candidates for the positions. Then it can automatically assemble into the entire protein complex structure. So this is the algorithm of the diffmodelers. So diffmodeler is able to build the high quality complex models using the traced backbone positions. Then it automatically combine each AlphaFold or predicted structure models into the large macromolecular structure. So this is the overview of the diff model. So this is these are examples. So we benchmarked a lot of different targets So this is a typical examples of our result. So we tried a 7.4 angstrom, 7.5, and 5.8 angstrom resolution map. So in general, so this density map, backbone position is really difficult to find. Then these rainbow structures, different chains are showing that's a native deposit VDB structures. Then it's also showing that's the original cryo-EM map. Then by using the diffusion map, it enhances the positions for the backbone position. So even the input map have a lot of noise data or strange low resolution map, but diffusion models enhance the backbone positions. Then in the process, the model also enhance the locally high probability positions using the LDP local dense point. It's kind of further enhance the backbone positions. Then by using these informations, so the modeler fit the AlphaFold input protein structure into the diffusion map. Then it generates these complex macromolecular structures. So as we know that the AlphaFold sometimes have a strange tail loop or extended loop regions, but we know that some core regions, core structure is very accurate. Then, the modeler can fit these core regions into the diffused map. So this is a comparison between the native structure and diff modeler models. So red is a PDB deposit structure. Then blue is a diff modeler models. So overall, it looks well fitted. So recently, we use these diff modelers to enhance the accuracy of the modelings. So one collaborator in the Nature papers, it's recently published in the 21st May this year. So they have these GPCLs and Aristines complex models. Then we apply the diff models. So they say that's the use of the new conditional diff modeler model suggested that the path of the helical backbone of the receptor indicating conserved kink in the TM transmembrane spans. So this is the actual examples. So GPCR positions. We can see the 7 helical structure here, GPCR regions. Resolution is 3.4. It's not bad. But using the diffusion models, it can enhance the backbone positions. Then we can see some helical pitches very nicely. Then these informations are very helpful even for the manual modeling. So by using the diff modelers, of course, it can generate the complex structure automatically, but also experimental peoples or experimental groups or manual modeling can be helped by the diffused map because it will enhance the backbone positions very nicely. Okay, so we also compare with the other existing methods. So we use the 2 different data sets. So 5 to 10 angstrom resolution data set is a kind of intermediate resolution data set. Then also we tried the 0 to 5 angstrom resolution data set. The metric is the TM score. TM score is a structural similarity between the deposited PDB structures and predicted models. So higher is better. So if the TM score is reached to the 1.0, so that means it's perfect modeling. Then we compare with the Phoenix fitting function, fitting program, and Vesper. So this is the original Vesper, our rigid body fittings. But this case, so Phoenix and Vesper, we use the original density map. Then another tool is Model Angelo. So Model Angelo is one of the most famous modeling tools for CryoEM. Then also we compare with the DeepMath. DeepMath is our modeling tools. So placing the backbone positions and connecting the dot, then build the structures, considering the opposed to structures and local amino acid type features. So as we can see that the x-axis is other methods, the y-axis the Diff modeler performance. So almost all cases, so we Diff modeler achieved better results, higher team scores. So interestingly, the best part, so rigid body fitting part is the same, but the input map is different. So Diff modeler using the diffusion models enhanced map. Then Diff modeler Vespa use the original map. The almost all cases, Diff modeler achieve much better. So that means the enhancing the backbone positions helped to fit the AlphaFold2 structure into the map very nicely. It's necessary to achieve the high quality models. So this is a 0 to 5 angstrom resolution map. It's relatively high resolution, but even at the high resolution map, so we achieved a very nice result. But some targets, DeepMath achieved better. So because DeepMath is a tracing method, so even the AlphaFold2 structure is not so accurate, but DeepMath can trace the backbone positions, then connect the backbones, then build out atom structures. So in that case, DeepMath achieve better result. Okay. Then, how can we use the Diff modeler in the SB grid? So Diff modeler code is also available in the GitHub, but on the SB grid, you can easily use these programs. So first, if you have this program, SB grid command line CLI, please type this command, SB grid CLI info Diff modeler. Then it will show the availabilities on the SB grid. Then if you are already installed, then it should show the sum star, then 2.3 installed. But if you haven't installed, please try to install the code. Okay. Then once you install this Diff modeler code on SB grid, so this is the command how we can use this Diff modeler on the SB grid. So first, please activate the SB grid shell, SB grid CLI shell. Then the user need to copy the models. So it's a deep learning weight file, Diff modeler CP based models. Then also you need to copy the config file, Diff modeler CP config. Then it automatically downloads the weight file, the parameter files to use the diffusion model. So after executing these two commands, so you will find these two directories, so best models. This folder contains the weight file, and this folder contains the config file for the diffusion model. Then in the config directories, there's a diffmodeler.json. So this file controlling everything, the diffusion model, diffusion part. Then please edit this path, model path here. So because we downloaded the best model here, so maybe home, in my case, gterashi, the best models, diffusion best.pth.tar. Then it's all set. You can use the diff model. This is a configuration. Okay, then once you install and copy the white files, then change edit the config files, then you can type this command, diffmodelers, then -f options is specify the density map files, then p options is specify the directories of your models, your atomic models. Then config is specify the config file, JSON files. Then put in the number of the control levels, then specify the GPU ID, then approximate resolutions. Then also you need to specify that's the PDB chain config file. So this file is controlling how many copies you want to generate from the input PDB files. So for example, this is a AlphaFold DB PDB structures. I want to use this PDB file to make the 4 chains, A, B, C, D, 4 chains. So in that case, in the config files, the first column is a PDB file name. Then I want to use this PDB file for the A chain, B chain, C chain, D chains. So first column is the file name, PDB file name. Then second to last is the assigned chain IDs. Then you can run the diff models. So this is actually examples for DB models. Then using this config files, let's try to fit this PDB structure into this 6824 MRC file. So if you have a different structures, for example, heterooligomer structures, you can add another lines. So let's say AAA.pdb, then you can specify the chain ID, chain EFG, like so. So this case is a tetramer. I have only 1 PDB files and 4 chain IDs. But if you have a heterooligomer structure, heterooligomer modeling, you want to do that, so please specify the different line, then add the PDB file names, then specify the chain IDs. That's it. Then after computing the diff modelers, so it will generate the predict result directory. In the predict result directories, based on the input MRC file names, it will automatically generate this ID name directories. The inside of this ID name directories, you can see a lot of different structure, but important files is this one. This one is the input file, of course, input density map. Then diffmodeler.cif. So this is the assembled model. Like this. Then also the diffusion.mrc. So this file shows the backbone enhanced map. So from the input density map, it generates this backbone enhanced map by using the diffusion models, then generates the assembled models, complex structure models. So these files give us a lot of information. So once you run the diff modelers, so please check the inside of the directory. Then actually, last week I tried to use the diff modeler on SB Grid using the different map and structures. So this is actual command I tried. So I used the 21843. So resolution is a 3.45. It's not bad. It's relatively high resolution map. Then I downloaded the AlphaFold database model. It looks like a lot of strange loop, but some core region looks very accurate. Then this is the input density map. Then this is the result. So backbone enhanced map with this. Then output structure looks like this. It's nicely packed, assembled the structure. So as a config file, I specified that I want to use this AlphaFold structures for A chain and B chain. Then it assembled into the homotime. So even at the 3.4 angstrom high resolution map, but diffusion map also provides a lot of more information. So for example, I highlighted the helix bundle structure here. There are some bump, maybe it corresponds to the side chains positions. But from here, diff modelers enhance the backbone positions. So it's nicely generates the helix pitches very clearly. Then these helix pitch positions are almost identical with the deposit native structures. So even for the high resolution map, if we want to carefully check the helix pitch positions or kinked regions, so diff modeler also help you to identify that these backbone positions very precisely, very accurately. Okay, then next topic is a cryo-read, so DNA, RNA structure modeling from cryo-map. So cryo-read is they are quite similar with a deep mass. So from the cryo-EM map, the deep learning unit architectures finding the positions of the phosphate and sugar and the backbone positions. Then using that information, it can trace the backbone like these lines. Then deep learning unit architecture also can identify what kind of base type are existing. So by using that information, we can assign the sequence to these lines, then build the 3D entire structures. So this is a cryo-read. So using the unit, it scans the density map, the entire regions. Then first, it tries to find where is the protein, where is the sugar, where is the phosphate, and the position of the base. At the same time, it computes the base type for its positions. The unique part is that the cryo-read doing this prediction twice, not only once. So because once we predicted the sugar phosphate base position predictions and base type predictions, we can cryo-read further improve, refine the predictions using that initial predictions. So this is a kind of unique architecture in the cryo-read. So we also compare how these two-step predictions improve the performance. So this is segmentation performance, how accurately identify the sugar positions, phosphate positions, and base positions. So in many cases, stage 2, second predictions improve the performance of the sugar phosphate base predictions. And also the base type predictions, yeah, it's not changed much, but C and G, cytosine and guanine predictions are improved a lot. So by using the two-step prediction, CryoRead enhance the performance. So we compare with the previous method, Phoenix. Phoenix has a DNA RNA structure modeling tools. So x-axis is the Phoenix, then y-axis the CryoRead, then backbone records. So how accurately backbone positions are predicted, percentage of the nucleic acid detected. Also, we check the sequence records. It's not only considering the positions, but also the type of the base type. So predicted the positions accurately, and also the type of the base are predicted accurately or not. So in both metrics, so clearly the CryoRead achieved a much better result performance than the Phoenix. Okay, these are examples. So CryoRead was published in the 2023, the Nature method. So these are reported the results. So resolution of 2.8, it's very high resolutions. Even the very big, large structure, ribosome structures. So we can build the structures very nicely. And also the DNA RNA position is a small part of the map. Only this part is corresponding to the RNA structure. But the CryoRead automatically identified as where is the DNA or where is the RNA, then build the structure. This is the CryoRead results. Okay, then how can we use the CryoRead on the SB grid? So the CryoRead is also available on the GitHub page. So there's a lot of explanations what is the parameter and what kind of parameter we need to change, like so on. But in the SB grid, yeah, it's very easy to use. So yeah, please first type the SB grid CLI info CryoRead. Then it will show these informations. So then latest versions is 11. So because last week, yeah, we updated the code. SB Grid teams updated the code in the SB Grid code in the package. So if you already installed the previous versions, maybe 2.0, please update the CryoRead code on SB Grid now. So it's 11, so it's totally different. It's much better code. Okay. So please make sure that you install the version 11 on SB Grid. Then to use the CryoRead, so first, please activate the SB Grid shell command. SB Grid CLI shells. They activate the environment of the SB Grid. The CryoRead have two functions. The first is if you have a sequence file, then based on the sequence, CryoRead can build the models. But if you don't have a sequence file, CryoRead assume that predict the base type, then build the entire structures. There are two modes. So if you have a sequence file, FASTA files, please use this command. So cryoread.main.py, then -f options specify the input density map, then -m best models, then control levels, GPU ID, and minus defines. So these options specify that if you want to define the Clara-Reed model using the Phoenix refinement architecture, please use this. Then Phoenix further improve the bond connectivities and remove the crash, like so on. Then -p options, please specify the sequence file. Then map resolution. So this is the examples of the input density map. 21051 resolution is 3.7 oxlo. So if you don't have a sequence file, suppose if you don't have a sequence file, please remove the -p option. Then put this no thick info options. It automatically finding the backbone positions from the map, then assign the base type. Okay, so this is actual output files. So in the predict result directory, you will find these subdirectories based on the input density map. Then there are several folders. But maybe it is good to see that this second stage detections. So because the Clara-Reed have a two-stage detection part, the second stage show the better performance to detect the base type and phosphate sugar base positions. So for example, this first 1, 2, 3, 4 MRC files, so it will give you the predicted base type with the probability values. I visualize on camera, I visualize these 4 maps using the probability cut of 50%. So counter levels 0.5. Then it will show that what kind of the base type are predicted at these positions. Then in second stage detection directories, there are another files. So phosphate probability, protein probability, sugar probability, base probability. So these 4 files are showing that the detected type of the protein or other structures. So I visualize using the 50% probability cutoff on the camera. Then protein probability showing that these orange, pink regions is corresponding to the protein regions. So input map is this. From this input map, it detects the protein regions like this. Then sugar position green position looks like this line. So U-Net deep learning detected these green positions have over 50% of the probability of the sugar. Then yellow is a base. So inside of this green ring, helical regions, U-Net detected that these positions are sugar. Then these magenta colored regions, so these positions are predicted as phosphate. So these second stage detections provides a lot of information about the DNA RNA structures. So then also the Clara-Reed.pdb. So this is the final models after Flatom modelings and the refinement by the Phoenix. They will show that these Flatom structures. Okay. Okay, so yeah, the remaining part is I want to advertise our servers. So because we are also providing the servers. So if you don't have a GPU card, or if you don't have enough resource, we are also releasing the code and also the servers. So please freely try to our Diffmodeler servers. It's completely same functions. So inputs the density map and uploading the alpha2 structure to here, they automatically assemble the model. So Diffmodeler sequence. So if you don't want to download alpha2 models, or if you don't have any atomic models, don't worry. So this servers automatically search against the PDBs and alpha database based on your input sequence files. So this is also a convenient way. Please also try. The CryoRead server is also available in our servers. So this is completely free. Please try. So currently, we are developing a lot of different tools. That we are adding the new tools to our servers, and also GitHub page, and also the SB Grid. So please try to use our tools. So Deep Math is available on our server and SB Grid, and Diffmodeler. Today I presented the Diffmodeler is also available. Then CryoRead. And last time I presented the DAQ score. This is a validation tool. So you have a protein structures and density map, but you want to validate, assess the quality of the models. So DAQ score provides the very nice evaluations. So please try these tools on SB Grid or our servers. So from here, algorithm, if you want to use the server, so please click the algorithms, then you can jump to the input job submission page. And also we recently start the YouTube channel. So Kihara Bioinformatics Lab. So we are explaining that on these YouTube channels, how to use our tools, then what is the algorithms. So it is very nice. So our students made the very nice videos. So yeah, please visit our YouTube channels, then try to learn how to use our tools. Okay, so I'd like to thank our lab members, and especially the Professor Kihara. He leads our project. Then also the Diffmodeler, diffusion modeling tools and CryoRead. So these two tools are developed by the Dr. Xiao Wang. Then Han Zhu, he developed the Vesper GPU fitting part. Then also, I'd like to thank the Kihara members. Okay, just thank you for your attention. So yeah, please freely ask any questions in chat box or any other ways. Also, the paper was published, the Diffmodeler paper was published last year. The CryoRead is available. And SB Grid is a wonderful tools to use our tools very easily. So that's it. So I'm happy to answer any questions. Thank you for your attention.
Thank you very much. That was a great talk. And I have a few questions here. You can put your questions into the chat. You can also click the raise hand button and your hand will raise, and then I can unmute you and you could ask your question directly if you prefer. I think the first question that came up was just along the way, or how you would rank the performance of these tools relative to other tools. I think it's clear from the slides you showed that like Diffmodeler seems like, okay, it's much more accurate than maybe Modelangelo or similar tools. what does the relative performance look like compared to other tools?
Okay, so uh, regarding the comparison with Model Angelos, so yeah, we know that if the resolution is higher than 3.5 or 3 angstrom resolutions, Model Angelos generates a very nice structure. But the map resolution is lower than 3.5 angstrom, then we know that the Model Angelo generates a very choppy, fragmented structure. But our diff modeler is even the map resolution is not great, but it can enhance the position of the backbone. Then it tried to fit the predicted AlphaFold2 structure into the map. So that's why the relatively low resolution maps, so diff modelers can achieve the very good performance comparing with the Model Angelo.
Yep.
Does it
Let's see.
answer the questions?
Yeah, I
Okay.
think so. I think that resolution differentiation is important, right? Because I think it's clear that once you get to a certain place, that's a nonlinear relationship in the sort of quality of output versus resolution, right? It tends to fall off very steeply with some tools. You reach a certain point, and then it's basically after that, it's very inaccurate. So that's an important distinction. Another question that we had was when using cryo-read, how do you interpret the confidence in the type of base assignment? Is it simple to tell which bases are competently predicted and which have a high probability to be mislabeled?
Oh, good questions. Okay, so uh, probability itself is kind of showing that the confidence. So if the predicted base type have a high probability, so that means that it's very confident of that base type. But considering the quality of the models, okay, yeah, currently we are working on the evaluation like DAQ score. DAQ score is also using the probability of the amino acid type. So maybe we can we can provide the confidence levels based on the probabilities. But short answer is that the probability itself is showing the confidence level of the prediction.
The confidence level of the prediction is based on the fit,
Yes.
and that is the singular metric of that. Right.
Yes.
All right. We had one other. Well, here we've got a couple. How long does it take diffmodeler to run on 1,000 residues? Or another way to ask that question: when would you recommend diffmodeler versus deep main mass, like the tools that have sort of
Okay. Good
semi-overlapping
questions.
functionality? Yeah.
Okay. So first, the deep main mass. Deep main mass is a tracing method. So backbone tracings. If the resolution is higher than 4 angstrom, then tracing works very well. But problem is that it's tracing the dot positions, so it takes time. So because there are a lot of possible possible tracing ways. So if the number of the residue is larger than 5,000, then it will take at least 24 hours. Then comparing with the diff modeler, it's very quick because it's not tracing. It's first enhancing the backbone position using the diffusion models, then rigid body fittings. So atomic models are fitted to the enhanced map. In general, 10,000 residues, less than 5 hours. It's very quick, but it depends on the quality of the map and volume of the structures. So if volume is huge, then you provide a very small piece of the structures, but you try to find the best fitting positions. So it also takes time, but it's less than 1 day. So typically it's computations should be done 5 hours. Yeah, huge one, 10,000 pages.
Yeah. So you would go to the diffusion model based approach for very large things over the threading, clearly. What is the biggest thing that you've done that you've run with the diffusion?
Biggest things.
Yeah, what I'm just curious, like what do you think the upper limit is in terms of, is it limited by your GPU RAM or is it limited by um how large, what's the largest structure that you personally have run through?
Oh, okay. Yeah, we already tried the 10,000, 20,000 residues. Yes. So diffusion part itself, yeah, it doesn't have a upper limit. Yeah, if you have a sufficient GPUs and computational times. Yeah, of course it will take time. If huge map, yeah, it will take 1 day. Yeah, but basically it's no upper limit.
Does it
Theoretically.
have does it have any knowledge of symmetry input in the process?
Oh, currently, yeah, we haven't implemented the symmetrical information. So just as it is, input map, to enhance the backbone position.
Okay. Interesting. Any other questions? Put them in the chat. Oh, we got another one here. For sidechain modeling in cryo-EM, is it more accurate to predict torsion angles directly from the density map rather than relying on AlphaFold derived structures? When would this be preferable? Is it also decided based on the resolution of available data?
Okay. Okay, torsion angles. So because AlphaFold models generates a very accurate torsion angles locally, then after fittings, it should be accurate than the tracing of the backbone positions. But we also know that the AlphaFold is not perfect. So sometimes the structures have a little bit different, or some shiftings happens, or domain orientation is not accurate. So in that case, current diff models cannot fix that problems. So it's rigid body fitting. So yeah, it really depends on the resolutions and also the accuracy of the AlphaFold model.
Right. Great. I've got one more question before we let you go. Thank you from the person who asked that question from Surabi. And it's just a practical one. What's the load like on your servers with all of these services and all of the different processes that people can run? Do people typically have a wait, or what's it like?
Waiter.
Yeah, I mean, do people typically have to wait before their jobs start, or is it what's the load like on your services for I saw all of these tools and I thought, wow, you must be running a small cluster there to support all of this.
Oh, okay. So yeah, it's technical question. So front ends, we have only one servers, but back end we have a lot of clusters. So we assign the 10 our local cluster machines, then using the queue systems, take the lot of job is running, you can get the result immediately. But if there are a lot of jobs, especially the heavy jobs, so like deep main mass for the 10,000 residues, then sometimes these jobs are submitted 3 or 4, then it occupies the CPUs and GPUs, then you need to wait for 1 or 2 days. But you can check the current status of the servers, how many jobs are running now. Yeah.
Oh, no, it's fantastic service that people can just log in and it's ready to go and they can just run their jobs right there. If they don't have GPUs or they just want to try it out and use something easy.
Yes,
It's
and
great.
also you can make the account. So there's some sign-in options here. You can make the account. You can keep your jobs in your own page. So submit the jobs, then close the browsers, but later you can check your jobs. So your personal page is available for the registered user.
Oh, fantastic. Well, that's great. That's a fantastic resource for a lot of very useful and computationally intensive, but super valuable tools. I mean, these are very, so, well, great. Genki, thank you very much for a great talk. Thank you, everyone, for coming. And thank you for rounding out the 2024-25 SB Grid webinar season. Everyone, keep an eye on the schedule for next fall. And if you've got anyone you want to hear from, definitely reach out. With that, we'll see you in September. Thank you.